So in this video, I'm going to talk about the cell cycle and mitosis. In some videos, they'll do them separately, but mitosis is a part of the cell cycle, so it makes sense to go on about both of them at the same time. Mitosis describes the splitting of the cell, of a eukaryotic cell, and the cell cycle is basically the life cycle of a eukaryotic cell. So when you think about a cell, let's just use an animal cell. We have one animal cell and we have our nucleus with our chromosomes, and it's just getting on with its normal life. But at some point, this cell gets too old and it needs to die. So what this cell does is it makes a copy of itself, okay? So the process of mitosis ends up making another genetically identical cell, okay? So that's very important. Mitosis creates an identical genetic copy, okay? And because of that, it means that if you've got, I don't know, this is a bicep muscle, and you want your bicep to be bigger, like that, you need more cells. So this is all made up of cells. The tissue is made up of cells. If you want more tissue, you need more cells. So the job of mitosis is to, or the function of mitosis is to repair stuff. So if one of these gets broken, you need to make another copy, an exact copy. So repairing cells by making a new copy, but also to grow cells. So you'll see it says the growth. This is the function of mitosis, okay? The function of mitosis is for growth and repair. It also allows cells, well, some cells, to reproduce asexually, okay? And that means they make genetic copies of themselves. So in some plants and in some fungi, they reproduce asexually and they just make a copy of themselves. However, this reduces the genetic diversity. So it makes species not very different. Okay, so this is mitosis, growth and repair, and in some organisms it allows it to reproduce asexually. But the cell cycle, as I said before, is the story of the cell. Okay, and I used to get this confused actually. I mean, I think it's easy to listen to somebody go on about it and it makes sense, but when you think about it, it didn't make sense. So okay, this is your cell cycle. And it's made up of 4 parts. Okay, you have the M phase, which is the mitosis, which is the splitting of the cell. Okay, you have a part called G phase 1, which is a gap 1 or gap phase 1. You have a process called S phase, which is DNA synthesis. Okay, sometimes seen as DNA replication. This is where DNA replication takes place, in this DNA synthesis bit. And then you have G phase 2, which is gap phase 2. Now, the overview is that your cell has its normal functions to make proteins and do its jobs. So in G phase 1, your cell is pretty much doing its normal thing. It's growing and it's making lots of proteins. When the cell realizes it's near the time it needs to divide, for any of the reasons I stated above, the cell creates twice as much genetic material, and it does that in this phase, the DNA synthesis phase. So now there's twice as much genetic material. Then it goes through G phase 2, which basically just does what G1 does, but a bit faster. So it makes its proteins and it grows, but a lot faster. In some eukaryotic species, it doesn't even use G phase 2, it just goes straight to mitosis. And then once it's done G phase 2, the cell splits. But because there's twice as much genetic information from here, you can have 2 cells. Okay, hopefully that makes sense. But this used to confuse me a bit because you get 46 chromosomes in a human cell. So I'm going, well, you have your chromosome here. So let's say at this stage, if I looked into my cell, I have 46 of them because that's what a chromosome looks like. And then you go through this bit and all these 46 chromosomes double. So it really confused me. Do you have 46 chromosomes here and then 92 here or not? And then it took me a lot of questions actually for somebody to give me the answer. But that's not true. A chromosome doesn't always look like this kind of cross that we see. It's not always like that. In fact, it's only ever like that one part. Here you have your chromosome, your cell, and your chromosome is made up of this one chromatid with a centromere inside. And that's what it's got there's 46 of these, but basically all the genes on, let's say, chromosome number 1 are there on that chromosome. Okay? Then what happens is it goes through DNA synthesis and all the information on this cell gets doubled. Okay, so we had our information in one strand here. Now you have a chromosome that has 2 of them strands. So this information is doubled onto one chromosome. So you don't have twice as many chromosomes. You still have 46. They just have twice as much material. Okay, I hope that makes sense. What happens in this stage, DNA synthesis, will be in another video called DNA replication, because that's a process that's quite complicated, or can be quite complicated on itself. So it's in a different video. So hopefully you understand the life cycle of the cell. G phase 1, doing its normal stuff, growing. When you go further into detail, there's certain cancer checks and proteins that check for cancer and abnormalities within a cell in G phase 1 around here and around here, okay, in G phase 2. But just for this video, we're talking about the cycle. So you've got your G phase 1, cell's getting on with it, starts to know that it's going to split sometime. So it copies twice as much stuff, cell gets on with it again, and then it goes into mitosis. That's the cell cycle, straightforward as that. So let's have a look in mitosis in a little bit more detail. Well, mitosis is split into 4 stages. So you've got one cell here, like this, and it goes into 4 stages. Okay, 1, 2, 3, 4. And through these stages, this cell here is going to make 2 cells that have that chromosome split in 2, but all the genetic material is the same. That's what's going to happen here. Okay, so we'll go through each stage. So our cell at the moment now has twice as much genetic material. Okay, so the chromosomes, this is the cell. This is a big nucleus just for ease of illustration. There's 1 chromosome. There's 2. It's been through DNA replication, so there's twice as much information on each chromosome. Okay, and this stage, the first stage, okay, is called prophase. This is the first stage of mitosis. And what happens in prophase of mitosis is that these chromosomes condense, which means they get more tightly packed. And that's because they know they're going to get split up, so they don't want to fall apart too easy. Another thing you have that goes on is some things called, I'll do them in red, some things called centrioles, some organelles called centrioles, start to move towards the poles of the cell. So there's 2 of them, and they start to move towards the poles of the cell. Okay, the opposite ones, the opposite poles. And these are going to make some proteins that come out and grab these chromosomes. As they move towards the cell, they start to create a set of proteins called spindle fibers, which start to come out of them. Okay? So they're in yellow there, they're starting to come out. And the last thing that happens is the nuclear envelope through all this phase starts to break down. The next stage, these chromosomes line up along the equator of the cell, so the middle of the cell. So we've got our two chromosomes, and they line up across the equator of the cell. Now our centrioles are at the poles, and our spindle fibres that was coming out of the centrioles are now attached to the centromere, which is the big round bit in a chromosome. So you've got your centromere here, and these spindle fibres are now attached. Because these chromosomes are aligned across the middle, the phase is called meta, which means middle phase. So pro means before, okay, or beginning, and meta means the middle. So that's stage 1 and stage 2. Prophase, the nuclear envelope breaks down, the centrioles move towards the poles, and as they do, some spindle fibers start coming out, and the chromosomes condense and get tighter packed, more tightly packed. In metaphase, there is no nuclear envelope, the chromosomes are aligned, and the spindle fibers are now attached to the centromere. The third phase is when we have our centromere, we have our spindle fibers, but this time the spindle fibers have pulled the centromere apart and split the chromosome in two. So remember, we said that we had our chromosome like this, and this side is identical to this side. Okay, so this side is identical to that side because it copied in the DNA replication phase. So now what? These they were together before, and in anaphase, these get shorter, pulling this apart. Okay. So now you have your chromosome being pulled away from the other half. Okay, so they're being pulled in this direction. Okay. This is known as anaphase. Anaphase. Okay. So the splitting of the chromosomes takes place in anaphase. The final stage is called telophase or telophase, depending where you're from in the world. And telophase means, tele means the end of something, like a telephone is, well, it used to be the end of a line, I suppose, it's not so much now. Okay, so that's the end. So when you start to learn your old Greek and old Latin, words in science start to make sense. And what happens is you've got your cells now are kind of splitting like this. So I've got, this is going to be one cell, this is going to be the next cell. Okay. The chromosomes are now in there, we're at the end here. Okay, we're at the poles of the cell, and now the cells start to split in two like this. Okay, and as it's doing that, these start becoming in their own little cell. So what happens is the nuclear envelope is formed again, like so. So now you've got two independent nuclei, okay? And then this cytoplasm divides, and you end up with two cells. And these two cells are genetically identical. So this splitting's taken place and it's made two genetically identical cells. In meiosis, it doesn't make genetically identical cells, so don't get them confused. In mitosis, makes identical cells for growth and repair. In some textbooks, you'll see another phase called cytokinesis, which is basically the splitting of the cell, but it's pretty much the way that you remember mitosis by 4 stages. PMAT. Prophase, metaphase, anaphase, telophase. It's worth noting now that our cell has a chromosome that's made up of one chromatid, exactly like it is here. Then it'll go through its normal life again, double itself, and then you'll have your chromosome with 2 chromatids, and it'll just carry on again. So you've got your prophase, your metaphase, anaphase, and telophase. Okay, guys?